Welcome to the GloBIAS seminar series. Today we start with a new mini-series on infrastructure best practices to build a bioimaging project starting from a user request. Our chair today is Kota Miura and our speaker is Asta Matura.
Asta did cell biology research during the PhD at EMBO, Heidelberg, and then I have an overlap during this period. And she was in the lab of Francois Nederlek, where there's heavily computational plus cell biology type of work. And then postdoctoral work was done in the lab of Mathieu Pierre at the Institute of Curie in Paris. Now she's the head of image data services at Euro-BioImaging ERIC, the European research infrastructure that provides open access to biological and biomedical imaging instrumentation and data services through over 230 imaging facilities across Europe. So she's currently representing Euro-BioImaging and the imaging community at the EOSC Association and its task forces. She also supports coordination of EU projects, including AI for Life and Founding Guide, and working on to coordinate provision of image data analysis services through Euro-BioImaging Nodes. So we expect that she will explain all these different types of activities happening in Europe. And please welcome Asta Matua. Thank you.
Well, thank you, Kota, very much for the kind introduction. And well, I'm very excited to be here and talk to you all about our activities and understand how image analysis is progressing in this world. Okay, with that, I would start to share my screen, and I hope people can see it. If nobody complains, I'm assuming that's a yes. All right. Okay, so yeah, so warm welcome from my side as well, everybody who is joining from whatever part of the world, because this is GloBIAS, so we're all global about it. And today I will talk to you a little bit about image analysis, about image analysis services that are currently being offered at Euro-BioImaging, as well as important aspects that connect us to communities and communities that can give input towards making these procedures more smooth. All right, with that, I will begin right into it. Of course, before we get into it, we would like to understand who am I and what am I talking about, right? So Kota gave a very nice introduction. Thank you very much about that. But let me tell you a little bit about the organization that I work for. So it's called Euro-BioImaging ERIC, or European Research Infrastructure Consortium. So we are a distributed European research infrastructure. You see these green marked countries, they are our member states. We have 19 member states, and this basically comprises 247 individual sites that are imaging facilities that provide open access to imaging technologies. That means it's light microscopy, electron microscopy, different kinds of imaging technologies at these facilities. And as a user, you can access these through us. This surmounts to around 120 plus imaging technologies, so the diversity is pretty wide. We are also a publicly funded not-for-profit organization, and like Kota also mentioned, that we provide not just access to biological but also biomedical imaging technologies. However, it's not just the instruments that what we are about. We are also about a people infrastructure, because you don't just get access to instrumentation, but you also get access to expert technical staff that can support your imaging projects. We can also provide guidance on using image data repositories and image analysis, and I will get much more into that in the rest of the talk. And we also provide training opportunities from time to time for everybody across the world. All right. So this kind of gives you a little bit of an idea of the technology portfolio that Euro-BioImaging offers. We go all the way from electron microscopy, molecular imaging, cellular imaging, organoids, organs, small animal, plants, and all the way to human imaging. We also are able to provide access to correlative or multimodal imaging technologies. Now, with all these imaging technologies, there's one thing in common, that they all produce image data. So that's why we also provide image analysis as a service. And this is rather a cross-cutting service that goes all the way from data that is produced on the molecular level all the way to human imaging. This also becomes a rather complicated and an interesting endeavor when we talk about correlative and multimodal imaging, where people have to work with different kinds of data coming from different sources. So it is a very highly important service as we see it. All right. How do people access these services? Well, I will briefly tell you about it, that we actually we can provide an initial consultation to users if they want to access these services. They can then put in an access request. We can provide scientific advice if that's what the project requires, as well as a technical feasibility that is provided by our nodes. So our nodes were one of these facilities. You saw a lot of pins on that map, so the 247 facilities that I was talking about. Eventually, well, you get a service. What does this look like for somebody who wants to do image analysis? So let's say you are somebody who have used a microscope to produce beautiful images, and you have your favorite question to be answered. You can come to our Euro-BioImaging web portal, and what can happen sometimes is when you realize that how complex your data is, it might not be as easy for you to handle all that data. It could be the fact that you have to deal with different modalities and so on, and you might need some help. Well, that is the case when you can actually reach out to us, and we can put you in touch with experts at one of our nodes who can provide you help with image analysis and as well as some trainings around it. So this is the kind of service that we offer. So we try to see of image analysis as something that we would like to share, which are not just isolated at the facilities that are part of Euro-BioImaging, but really opening up this highly specialized technology for the rest of the world. All right, so let's imagine a scenario to understand what are the kind of services, what are the kind of support that you can have. So you have produced some really nice, valuable, let's say large data, image data, and then you realize that it's complicated for you to analyze this. Well, that's fine, no worries, you come to us. We are also, in fact, able to provide now image analysis as a standalone service, which means that you could have acquired your data anywhere. You don't just have to acquire data at a particular facility and get support for analysis from that specific facility itself, but you could be producing data at one facility and asking for analysis support in another facility. So currently these services are being offered by various facilities that are part of Euro-BioImaging. They are currently situated in 12 different countries, and support it can vary depending on the need of the user. You could want to have image data handling pipelines for large data, denoising, registration, segmentation, you name it. There are many technologies that one could require. What is also fantastic is a lot of experts at Euro-BioImaging Nodes. They actually are also maintaining and open tools and libraries for image analysis. So you can get specialized help if you want to, let's say, track cells. You want to have support in using TrackMate or the latest versions of the latest softwares like tissue maps or something to look at your spatial omics data. So all of that is possible because we have a lot of these people who are developing these open tools and repositories that are part of Euro-BioImaging. All right. What is our objective while we're trying to do this? So first of all, let's talk a little bit about what happens when you provide image analysis as a service. So there is a decent impact of these services, right? First of all, we get to share the expertise that not everybody has access to. There are lots of larger imaging facilities that are producing a lot of data, but they lack the expertise in terms of image analysis. So we want to be able to help those users as well. On the other hand, from an image analyst point of view, you get an increased visibility because as an expert, you are working together with external users, working on different projects, and all this opens up an interesting possibility for you to engage in different scientific and technical solutions. Another thing that is quite impactful is that the way these services work, they do promote fair image analysis practices. And again, fair is findable, accessible, interoperable, and reusable. Because there is large amount of data often that needs to be well annotated and it needs to be transferred to the analysts that are sitting remotely somewhere else. So, and a lot of times the analysis pipelines are also then made available to the user so that they can access it, run it in their own time. So this really is a way to promote practices that make the analysis pipelines also a little bit more reusable, or at least we are supporting that this actually takes place. However, it doesn't come easy, does it? So through a lot of expertise from our nodes and a lot of conversation that we have, we actually have a dedicated expert group in Euro-BioImaging where we talk to the image analysts regularly, trying to understand what are the challenges that they face in terms of service provision? Because this, again, for an image analyst crowd, like through GloBIAS, I think this is a very interesting consideration. So it could be simple technical things like having troubles in data transfer because the data can be really, really large, right? So we try to share best practices around what are the best ways of transferring data. We also talk about how to make their services more visible, because oftentimes you would have image analysts in smaller facilities who might feel isolated and their services might not be very visible. So we are also trying to work and trying to understand how to make these services visible to a large potential pool of users. Another very interesting consideration that came up in many of our discussions, and this is again me sharing our experience from Euro-BioImaging side, is the estimation of effort that is required for service provision. Because from a bioimage analyst side, you would look at a service request and you might need to estimate how much effort it would be for you to actually work on the analysis problem. And this can be sometimes difficult because, you have some smaller data set and you have to extrapolate. And a lot of times we've seen image analysts having a slightly difficult time to estimate that. However, there are ways in which they're also trying to deal with being able to give a better estimate to people who want actually access to these services. And another rather important and let's say something that ties not just to the current work that you're doing now, but also towards a future where you can think of a good career progression is credit attribution. As a bioimage analyst, if you are working on a problem, what kind of attribution do you expect? If it's a pay for service, you still would, it depends on how much you have contributed to the specific problem itself, and how to agree on that with the users is always a little bit challenging, or that's some considerations that we have to keep in mind when people are starting to formulate these services and trying to provide them and interacting with the users on a regular basis. So again, some of these conversations, we are always discussing about these things. If you have any ideas, well, please reach out to us. We are always listening, and we are working currently with our expert group to come up with some guidelines or some suggestions on how to deal with some of these challenges that image analysis service providers can face. I'm sure we're going to come back to some of these in the discussion, so keep your questions ready, right? Okay, so this is a little bit about the process of providing services. And here I wanted to give a shout out to who are the people who are providing the services. So you've seen this map briefly before, right? It gives you an idea of all the facilities that are providing their services through Euro-BioImaging. And you will hear more very concrete examples in this seminar series itself in the next 2 months. So in August, you will hear from Anna Klenn, who's part of our Swedish NMI node, and she will also share some lessons from managing such a facility on a national level, as well as in September, you will hear from Jean-Yves Teneves in the same series, who is part of our France Bioimaging node, and he will also share his experience on a different structure of an image analyst node in France. So, well, stay tuned, that's it. All right. So now that we talk a little bit about the services, the next thing that I wanted to get out of the way was FAIR. Well, this is an acronym that I don't think of an acronym anymore. I feel like it's a word that's part of my life. So what we're talking about most of the times is, well, making the services that we are offering findable, accessible, interoperable, and reusable. And this is true not just for the services, but also for the data itself. Because as an image analyst, if you have tried to use data, you need to have some level of fairness to that data involved. Otherwise, it is impossible for one to be able to make sense of that data. So thinking about how to do those things is actually part of the whole providing image analysis as a service, if you think about it. So this is where I wanted to come to. So when we think of an ecosystem of fair image data and services, I already touched briefly upon providing fair image analysis services, how they are being provided through our nodes. But now I would like to take you through the other parts that we think are quite important in order to provide image analysis services. So this basically goes through, well, some kind of tools that enable this, fair data, like I was referring to briefly, and the role of larger and wider community in order to enable us to do this better. So, well, first let's get into some of the fair tools. I have already referred a little bit to the fact that, nowadays with the most advanced imaging technologies, the image data is becoming larger so much so that I would argue that the distinction between data management and data analysis is almost blurring, because you need to do a good level of data management in order to do data analysis effectively. And for that, we also need certain tools that enable us to do that. So the couple of examples, I tried to pull together a couple of examples in that direction. So one of the things brings me to a currently community-driven file format called OME-ZAR. I love to think of it as a Google Earth for image data, where you can have all the resolution you want, but look at it at a low resolution, but zoom into it and find all the lovely details of your images. So what we are trying to do is also trying to support this community effort around OME-ZAR through tooling. So this brings me to the development of tools and workflows for image data management and analysis. So please feel free to have a look at our GitHub page. You will find all of these different tools that include basically some little let's say services, like if you want to convert your data to OME-ZAR with a parallelized format, or if you want to define workflows which work directly in ZAR, that is also OME-ZAR, that is also possible, as well as the OME-ZAR pyramid, which is a Python library for working with ZAR data. These kind of tools we're trying to offer them to the community so that we can fill a gap if that's what is enabling people to be able to deal with this large amount of data. And these tools are also available as content packages, so feel free to check them out. And we do encourage people to use OME-ZAR and support it as a file format. If you need some training, you can also follow these links, and I will share my slides later, so do not worry, you will have all these links as well. So there are some training materials that can help you get a little introduction into the NGFFs or the OMSR format. And I think you've also had a couple of talks within the GloBIAS community, as a lot of people are supporting, a lot of image analysts are also supporting tooling around the OMSR format. Another tool that we also think of it as an enabler here is the bioimage analysis desktop, or the BAND desktop. So this is basically an HPC supported remote desktop where you can perform image analysis. Now, please feel free to use BAND and try it out. There are various tools that are pre-installed and available for you to use directly. So these tend to be the most popularly used image analysis tools. However, we are maintaining it regularly. So if you access BAND and you would want some tools to be added, which could be interesting for the community, you can also just reach out to us, and we will see what we can do in order to include your tools so that we can also spread the tools a bit further into the community. And here I will give a little shout out to image.sc, because you can find us on image.sc on the forum. And if you have any questions, especially if you want to use a BAND for trainings, this is also possible. In that case, we block the online, the desktop for training procedure, for training courses. So please reach out to us, and we would love to see this resource being used by the wider community. All right. With that, another interesting tool for the more advanced image analysis, well, nowadays is AI-based image analysis, machine learning or deep learning methods. So a lot of these developments that I'm going to talk to you briefly about were developed as part of the AI for Life project. So in this project, basically, we wanted to work towards making the cutting-edge AI methods easily usable by life scientists. For this, I would encourage you to check out the BioImage.io or the BioImage Model Zoo. Now, this is a repository of pre-trained image analysis models that you can just go there, you can check it out, you can use them on your data as well. And a lot of the efforts within this project were made to extend functionality of the bioimage model zoo. Additionally, what we've also worked on is to get AI-ready datasets, because it's not just the model that is often enough for you to understand what you want to do. What you want to have is you want to also have access to AI-ready datasets that tell you what are, first of all, the data that the model was trained on, and also give you an opportunity to use that data to train other models, maybe your own model. Now, these datasets find their way into the Bioimage Archive, which is a dedicated repository for image data that we encourage people to use, because that really makes it really tries to convert image data into a resource that people can use globally, right? You have well annotated data stored in this repository. And, I always like to think of things like, think of bioinformatics. How does that work? You have repositories that store all this fantastic data, and it enables bioinformaticians across the world to use that data. Well, we're hoping and we're having same aspirations for the Bioimage Archive as well. So please do check it out if you do not already use it. Now, one thing that is currently ongoing in the AI for Life project, and I'm going to give a little introduction on that, is challenges. So for all you image analysts out there, we currently have 2 challenges ongoing. So they are both denoising challenges. There is one on supervised denoising, and another one on calcium imaging, denoising of calcium imaging. So please check it out. It's on the Grand Challenges website. Here are the links that I'll be happy to share. And well, let's see who ends up on top of that leaderboard, huh? So looking forward to seeing you all participate in these activities. All right. So now that we've covered a little bit of the specificities of AI methods and image analysis methods for, well, biological imaging. I just wanted to bring across this little example to you because it is fascinating to think that image analysis is not just specific to bioimaging, right? There are other domains that also use imaging as a methodology. So in this project called the Oscars Fiesta, we are trying to explore how we can extend the bioimaging methods to different domains like astronomy or environmental sciences. So this is, again, like really trying to expand the horizons of what we want to do or we want to achieve as bioimage analysts in a way. This is, again, feel free to check it out. And we also have an expert group around this work. It's an expert group on fair image analysis workflows. And in this particular project, we're really working on how these workflows can be shared between even different domains. So again, this also gives us an opportunity to structure our workflows in a way that it's not just understandable by people from within the domain, but across domains. Really valuable interoperability work is being done in this project. So just to open up our horizons a little bit. All right, so we talked a little bit about the tools, but we also had other things on our wheel that is enabling our fair image data analysis, right? So the next thing I want to bring to you is fair data. Now, when it comes to fair data, it's one of those things that there's a lot of information. There is a lot of things going on. And I think at some point of time, it is important for people to be able to get that information. And what we are trying to do is we are trying to support people and making that information readily available. So we do this through image data stewardship. So we have data steward support at Euro-BioImaging, and we are very closely collaborating, first of all, with image data repositories, because this is then becoming quite a cornerstone of making image data available to the wider audience. We are basically trying to be the bridge between the users and the repositories and enhancing the data influx into the repositories, because the more data we have, the more quality data we have, more of these analysis pipelines, potentially reusing existing data, we can envision and actually have. So we are closely working with Bioimage Archive. I've already mentioned that repository, but also other repositories like the ITR, Empire, which are data repositories, but also a preclinical data metadata repository called PDART, which is maintained by Euro-BioImaging. All right. So again, like I said, so to promote practices around fair data, which eventually will make our analysis a bit easier, we also try to have regular training events. We try to work together with people from our nodes and have online workshops that anybody can attend. And a lot of this material is made available. So please feel free to check out our YouTube channel, but also our webpage, where you will find the materials on how to make image data fair. The most recent edition of the Guide to Fair Bioimage Data was focused on imaging facilities. So we're really trying to understand, because that is one place where a lot of image data is being generated. So we think of them as multipliers of a lot of these good practices. So we want to understand how to ingest data, how to get data from instrument into data management systems, and then eventually through analysis or after analysis into repositories, so that we can really close this loop and be able to track the image data through the whole process, through the life cycle of the data. Now let's talk about little resources that can also help you. So yes, you can sit through the talks and listen to that. But in addition to that, there are little things that can make your life easier, and not just your life easier. You can feel free to share it with people who come to you with data that is not very well organized like us. So we actually have a template data management plan, which is a bit tailored to imaging facilities and imaging use. that you can access, share, reuse if you like. This is basically helpful for people to plan their imaging experiments even before they've actually generated data. And actually, in many grants, this is almost a requirement nowadays that you should have a data management plan before you start to actually work on your grant, so that you know what kind of data you are going to produce, how you're going to use it, where you're going to store it, how you're going to analyze it, and where are the analysis results eventually going to live. So please have a look there. Then, of course, we get a lot of these questions about, if I want to submit my data, where should I submit it? That's an interesting question. It's not always straightforward where the data goes. But if you want a little help in that, we have a little decision tree that you can follow to understand where your data goes. So really trying to help people find the suitable repositories. And if you want to follow a step-by-step deposition recipe, you can go to the FAIR Cookbook webpage, where you have a step-by-step process that one can follow. And again, this is useful for people, not just who are producing the data, but also people at the facilities who can distribute these good practices amongst people, amongst the users, so that we're really building up this FAIR image data resource now. Yeah, there are some publications, so you can have a look at that later. Now, coming back to our wheel. So we've tried to figure out how to provide FAIR image analysis services. We looked at some tools. We figured out how to, let's say, make our data a little bit more fair. But now let's have a look at how a community effort can actually help in this. And I cannot stress enough how important all the work that is ongoing in the community is. There are various community initiatives, whether they are technical, based on certain technologies, for example, the volume EM community, or dealing with certain aspects of image of data, like EDAM, which is specifically working towards ontologies, or infrastructure platforms like Galaxy, or data management platforms like Omero, and so on, or more people infrastructure, or communities like GloBIAS or ESME or Quaraplini for quality management. And I will come back to global bioimaging later. But for us, it is very important that a lot of these developments that are being done in these communities are not done in isolation. Hence, we focus a lot of our efforts in coordinating and communicating amongst these community initiatives as well, by talking to each other, by making sure we are present in many of the working groups, so that whatever is being developed, whether it's at our nodes or at an imaging facility in Brisbane. So we just want to make sure that we are talking to each other, so that we can benefit also from each other's knowledge. Again, coordination with technical as well as community initiatives is very important for us. This brings me to one of the projects that I wanted to tell you about, which is really coming bottom up from the community, but trying to answer something on a global level. So this project is called Founding Guide, or Founding a Global Image Data Ecosystem. Yes, we do have big plans here, right? So what we want to do is we want to have establish a coordination among global repositories, open image data repositories, all the way from Europe to Australia to Japan. So these are our project partners. We have partners in this project from Europe and Australia and Japan who are owning these repositories, and hence can work towards having a layer of interoperability on top of their repositories. And this is important because one way or the other, we're kind of moving towards not having to move a lot of large image data, but still being able to access it, or at least understand that it exists in another part of the world. So it's the discoverability of the data that we are also wanting to focus on in this project. While we are also developing some technical solutions in the project around suggestions or on ontologies that people should use, the structure of metadata that the repositories can have. But a very strong component of the Founding Guide project is community, because we want people to be aware of the things that we are developing in the project, but also understand what people need and see what they are developing so that we can also learn from them. And to do that, we have, we've planned 3 events in this project. The first one already took place last year in Japan, which was a great success. We were together with Global Bioimaging in Okazaki, and we had a very global participation, including with many people from GloBIAS as well. And this is now an invitation to you all, because this year we are going to be in Australia, in Brisbane, and our event is very conveniently sandwiched between 2 other events, which are longer and larger, which could be interesting for you also. We have the International Data Week, as well as the eResearch Australasia meeting. So for all those who are interested, the registration is still open. Please have a look, and please do join us in Australia, especially if you're in the region, which, given the time, I hope you're not. We would be more than happy to have you there. The idea is that we talk about how to share image data really across boundary and have solutions that can enable this to make image data more interoperable, and eventually thinking of a federated image resource. All right, so this was a little bit more technical, but let's come back now to global bioimaging. I wanted to bring this up because global bioimaging is really a network of cutting-edge bioimaging facilities and research infrastructures across the world. And a lot of the work that we are doing in Founding Guide is based on the connections that we had because of global bioimaging. So this, to me, fits as a nice rounding story of having the connection, having the idea, writing a project, and then being able to work on certain things together. And this is where the value of community also comes in. In this community initiative, the idea is to cooperate internationally and propose solutions that are amicable for everybody, that everybody can use. So we're really trying to address things that affect the global community here. The focus here is, of course, on imaging scientists, which include image analysts as well. So as you can see, there's lots of research infrastructure, regional ones, that are part of global bioimaging. I hope you all are also part of one or the other of these research infrastructures as well, or part of global bioimaging, as they have very relevant working groups. So they have 4 working groups that are running currently. There's one on career paths, again, something super relevant for both imaging scientists and facilities as well as image analysts. There's one on training, one on data management, and again, very relevant work here ongoing in terms of how to even think about managing this large data that is nowadays being produced in various facilities, as well as how to measure impact of our imaging facilities, because this is something that we need to do on a fairly regular basis to understand and to get across what is the impact of the services that the imaging facilities provide. With that, another little advertisement is that the Global Bioimaging is currently running through its Career Paths Expert Group a spotlight series. And the next talk is by none other than Beth Simini about how to befriend or become your friendly local bioimage analyst. I'm sure a lot of you can learn a lot from this talk, and I'm really looking forward to this one. So please feel free to register and join Beth in understanding, well, how to become your friendly local bioimage analyst. All right. With that, I wanted to tell you about another opportunity that actually comes through global bioimaging as well. So this one is called Imaging for All. This is a Wellcome Trust funded project that is run by Global Bioimaging that aims to provide people from low and middle income countries, the imaging scientists from these countries, to get access to imaging facilities or training across the globe. Now, what's interesting is that we've already had a very successful first open call of this project, but what's probably very relevant for you all is that the second open call is here. So you can put in your application until the 15th of August. You can, like I was mentioning, you can get grants for accessing imaging facilities if you like. You can also get a pro grant, which is to access quality managed imaging facilities, or training grants for you to attend training events that are relevant for your career progression and to bring back expertise to your imaging facilities as well. So please have a look. If these apply to you or your colleagues in LMICs, please spread the word. All right, I think this brings me to the end. And well, I'm just going to say thank you for giving me this opportunity to talk to you all. And I am very happy to take any questions if they are. I will be sharing my slides. So all that information that was unloaded on you, you will find the links, and I'll be happy to answer any questions now. Thank you very much.
Thank you, Asta, for a great talk. If you have any question or any kind of topic, so there are many explanation about different types of activities. And then if you have any question for one of those specific ones, it would be great to have one.
Kota, maybe I can read out the one question
Yeah.
we already have in the Q&A box.
Oh, okay, yeah.
So Richard actually would like to know if a user makes a request for image analysis advice, how do you decide where to direct them?
Oh, thank you, Risha, for that very interesting question. So currently, our process is such that if a user request comes in and they know which particular facility that they want to go to because they know of the specific expertise there, then they can apply to a specific node, specific facility, and we are basically redirecting them to that facility. However, I think your question applies to the cases where people do not know which facility they want to go to, but they only have an image analysis request. In this case, we moderate the request, and the current method of doing this is by asking all of our image analysts, like it's an open request to all of the image analysts, and they can then put up their hand if they are able to take up the project. So I contact all the nodes that provide image analysis as the service. And this basically also gives an opportunity for the project itself to get a wider visibility amongst various image analysts rather than maybe asking only one person at a time. So this is the current methodology that we use. And if we know that they have specific requirements that are met at a particular node, because again, we're also quite closely in touch with the services that are, or like the expertise of our various nodes or the facilities or the image analysts, then we can also direct them specifically if somebody wants a peculiar, like if they want to work on marine organisms that have been imaged under X-ray imaging. So these kind of things, if it's very specific, we can direct them. Otherwise, it would be an open call, and then the opportunity would be really for the image analysts to show interest. And then again, then comes the whole process of technical support and the technical review where they would probably need to discuss if it's possible or not. And then again, this gives us an opportunity that if it does not work out with the first image analyst, then they can quickly come back to us, and other image analysts have also already seen the request and can put up their hand in case required. I hope that answers your question.
So there's another question from Lior. Are you offering grants for maintaining or developing open source projects relevant to the bioimage community?
That's an excellent question, an excellent point, and I wish the answer to that was yes, but it's not, unfortunately. I would love to have that. And so most of the times we would like a lot of grants that we are able to give away are often defined by the grants because it's not our core money, but we are also applying for grants and then cascading them in most of the cases. So a lot of these requirements come from who the actual funder of grant is. So we need to follow those. And unfortunately, currently, we don't have anything on maintaining or developing open source relevant relevant tools. However, this is something that we keep a close eye on. So for example, the Oscars Fiesta project that I was talking about, the image analysis across domains, that was a result of one of these open calls. So it's not a project, it was not a grant that we were cascading, but it's a European project that had this open call. And anybody could apply, so did we. But we also know many of our nodes applied and then they got some funding there. So we try to keep an eye open for what are the opportunities and promote those. But from our own budget, we are not able to support these activities yet. I mean, this might change if we get a grant going in that direction.
Good. So I have some question about or some kind of request. So you said in part of this fair image data services, you said data management and data analysis is becoming somehow one big issue. Of course, I mean, so if my understanding is right, you're trying to say that there are a lot of these research with very data-heavy analysis going on, with huge amount of data, with very complex multidimensional data, with multi-channels. And then, so that in those cases, how do you actually manage those data? And analysis becomes a parallel problem. How do you actually compute all those things in a very efficient way and so on? But on the other hand, I think, yeah, so 99% of people, so life science people are not really having that problem. So, and then it's mostly a much, how to say, more basic problem. So, and then it's not even, they don't need, so many people do not need to access high performance computing systems and so on. But I think most of the people, if you see actual biology papers, what people are struggling is with really very basic image analysis, right? And then so that if you try to focus as a Euro-BioImaging to focus on something very, only small fraction of people in top institute are worried about, I think you might lose majority of life scientists who are actually struggling now. And so I'm wondering if there's any kind of discussion about this is going on in Euro-BioImaging.
Yeah, I mean, so first of all, absolutely. I mean, frankly, the top 1% of institutions that are really good at image analysis don't really need Euro-imaging either, for that matter, right? I mean, there are various reasons to be. I mean, I'm happy that they're offering their services, but I guess it's also we really try to make sure that we are talking to everybody and with everybody. And let me answer. I mean, let me try and answer a couple of things that I picked in your comments. So first of all, I do agree that when you have a very large project where you're producing very large data, you probably are already thinking about doing data management, yeah? Because otherwise it would just not work, right? So if you are taking a whole brain and like slicing it and like you want to do EM on it and then put it all together, you probably have a data management plan. You probably have dedicated data managers, you probably have data annotators, you have things in place because that usually is taken care of. And I understand that most of the problems that our daily scientists face, they are rather, smaller issues. And I totally agree. And this is one of the reasons why we are trying to, the kind of solutions that we're trying to offer. And again, here we are also always listening. Yeah? I mean, if you have ideas, more than happy to hear. But from our side, we want to make sure that we are doing things like supporting OMISAR or just making something like Band available to people where people can just log in with their Google account and click around and use a tool that is already installed, and they don't have to worry about installing one, right?
Yeah, yeah.
This is one of the reasons why we want to make sure that we are not forgetting, let's say, the large in number, but smaller in data volume sort of people.
Yeah,
And I'm
yeah.
glad you brought this up because so one more point is that we were talking with so in this expert group on image data, we were talking to the bioimage analysts there, and a lot of them also say that a lot of the problems that they're able to solve can also be done in this call for help or like a consultation phase, yeah? So maybe for a lot of things, people just need a little push, a little answer, and of course, image.sc is a great place for that, but yeah, forum fear and so on and so forth, it can happen. So I do think that we need to make sure that we are catering to everybody to a certain degree. So we try to do that, but I'm more than happy to hear some ideas or comments or discussions on this.
Yeah. So my idea is that somehow there needs to be some kind of marketing. So that so I have gut feeling. Yeah, if I see those all these journal cell biology papers or development papers or journal cell science papers, then as I said, so that I would say 99% of people are not doing data intensive study, but it's really image data in daily life. You have to really deal with somehow, and then somehow this analysis of the actual topography of image analysis problem is not really clear. So maybe Euro-BioImaging and global bioimaging can do something like this, so what is the actual, the issue, yeah? That you have to handle as a global network or Euro-BioImaging network. And then you might be able to gauge actually quantitatively what should be done. So because my kind of idea about this, because you can systematically do this because you have network, right?
Yeah, no, absolutely. But I mean, in a way, so correct me if I'm wrong. So there are also quite some efforts from GloBIAS on this, right? I mean, in terms of mapping the landscape, there is this regular survey that takes place, right? So in terms of trying to define what are the problems that people need support with, I think we do this kind of surveying of the landscape from time to time. So I think that is.
Yeah. So okay. So sorry, interrupting, but not about the demand of the support. But you can analyze this from papers. So what type of image analysis people are actually dealing with, then you can just make a survey just by accessing all the papers and say, okay, so that it's like this and like this. I mean, that's one way, but I don't know about what would be the best efficient method for doing this. But how to actually beyond the gut feeling that I just talked about, 99%. Yeah, but I think it's possible to really draw a more precise picture of what is actually needed, and that's what I think. It's always attractive to go with the high end and then do something really advanced and then make challenges, but then this might lose a lot of these actual scene of the image analysis. That's my kind of comment, and then also whether you guys are actually recognizing this type of problem. So that was my kind of a comment. And any other questions?
When people are thinking about their questions, I can also comment on what you're saying is that, no, for sure.
Okay. Yeah.
We're thinking about this because, again, like you said, that most of the times it depends. Like a lot of these things are based in context, yeah? So if your context is that you have access to everything, then maybe your needs are different. But if your context is that you don't have access to the cutting edge image technology, then maybe even a confocal is great for you, right? So I think it depends on the need. And most of the times, most of the imaging, maybe having a good enough confocal will solve it rather than going for light sheet and whatnot. I guess this is one of those things that we always keep in mind, and that is important for us, mostly because we do have a diversity of nodes. Yeah, we have nodes all the way from, I don't know, EMBL to Portugal to Sweden to Paris to Bulgaria, right? I mean, so it is quite diverse, and we do very strongly wish to serve them very well and equally. So support them in this, yeah.
Good. There's no question?
Ashta, I would have a short question, because at the beginning you mentioned that you are also have a group working with, on a summary of the challenges people are facing when actually offering image analysis as a service, and you briefly mentioned the problem of credit attribution. Are you there looking at something similar as we now have for imaging facilities? Because I think that is something that is really lacking, and it would be brilliant to see something like this.
So, I mean, that is an excellent point. I mean, we haven't thought about including specifically that aspect. It was just one of the aspects that comes up in discussion. But I think it would be quite important to address it. And you're right. One way of doing this could be to think of like an extension, or making sure that the aspects related to image analysis are also considered when you think of the guidelines for facility scientists, right? I mean, then maybe it's kind of covered in a vague-ish way, but if we think that we need some more additions to cover the basis for image analysts, I think it would be quite important to have that conversation going, and we can most certainly do that. Because again, it's one of those things that comes up in conversation, be like, oh, but you're paid for your service, and why do I need to put you as a co-author, right? And as if people don't pay their PhD students, but okay. There are somewhat clear ways of doing it, but I think, I don't know, maybe it's a movement, it's a lack of awareness, and a lack of requirement in certain cases, because we have also heard of facilities where they are either not allowed to be on research paper, or not supposed to be on research papers. But I think that's a very specific scenario, and we can't generalize that to everybody and not have recommendations in that direction. And we should treat certain things also as exceptions, I would say. But an excellent point, yeah. We should definitely.
Okay, so the time is almost up. And then thank you, Asta. And then we have a next seminar coming on 27th of August. Anna Clem will talk about behind the scenes at SciLife Lab. I mean, she's going to talk about probably, the actual what they're doing with this help and so on. It's pretty interesting if you are interested in this direction. Okay, so with this, we'd like to close our session. Thank you very much for attending, and see you next month. Ciao.
Thank you, everybody. Bye-bye.
Thanks, everyone. Bye.